Good afternoon. In the interest in staying on time, I'm going to start with our next presenting company, Fibrocell. My name is Ted Tenoff. I'm a managing director and senior biotech analyst at Piper Jaffray. Fibrocell is an autologous fibroblast cell therapy company. And here to discuss this story is John Maslowski, who's a senior vice president of scientific affairs. John, thanks for being with us today.
Thanks, Ted. Thanks for having us.
So we're going to skip on using slides and continue to do the sort of fireside chat format, if that's okay, the Q&A. And if you could start us out by describing Fibrocell's technology.
Sure. So as you just mentioned, we have a platform around the autologous fibroblast cell. So that falls into two buckets of products we're developing. The first is a non-modified, what I would call cell therapy version of an autologous fibroblast that's used for treatments, and we'll talk a little about the indication shortly, but we're in a phase 2 program in a vocal scarring condition for that particular product category. When we manufacture these, we manufacture them in our GMP facility outside of Philadelphia. It's GMP inspected for several years now, part of a BLA package we filed some years ago. But in that process, we collect cells from the patient directly from post-auricular biopsy punches. They're dermal punches. They're sent to our manufacturing facility, expanded ex vivo in our lab. And then they're expanded in short passage up to a large population, which is in totality then cryopreserved for use. Then they're thawed and washed and shipped overnight, fairly typical cold chain pattern, back to the site the next day, and then for autologous administration. The other buckets of products that we're developing off of this technology, however, is in conjunction with Intrexon, if you're familiar with them. And what we're doing is we're taking these cells and we're gene modifying them with monogenic targets to treat rare diseases of skin and tissue, and also some work we'll be doing in joints shortly. And this is done at low passage. We transduce the cells using a self-inactivating Gen 3 lentivirus, and then they're expanded from there. Since it's integrative, we then expand the cells after they're transduced, and then we collect that bank of cells that can be used back for patients. So two approaches.
And why is the fibroblast so important?
So in our particular disease category, we're targeting skin, connective tissue like mucosa, and also in the future be looking at connective tissue and joints. And in all these tissues, the commonality is how does the structure of these tissues form? And they're formed by fibroblasts that produce extracellular matrix proteins like collagens and elastins. So they reside in the area where we would like to administer these products. And unlike keratinocytes, which turn over quite often, they have longevity in that area. So we think that not only are they a great target, they're also, if you've all done cell culture, which I'm sure a lot of people have, when you grow cells, fibroblasts are quite easy to grow in comparison to, say, a keratinocyte, where you tend to be contaminated by fibroblasts. So it makes us for a more consistent manufacturing process, which we've done quite a bit of over the course of time, and it really is targeted to those areas for disease.
Okay, great. You mentioned the phase 2 study that you're currently conducting for vocal cord scarring. First, maybe if you can, tell us a little bit about this affliction or this disorder, and tell us about the product specifically, how it's addressing the condition.
Sure, good question. So vocal fold scarring, there's numbers of conditions that lead to this. So if you think of your vocal folds, how they work, they vibrate as air passes over them. They generate voice from the noise they vibrate off of the mucosal surface of the vocal fold. From overuse, from damage, from atrophy from age, and even from surgery or radiation treatments, there's a lot of conditions that can cause these to thicken from scars. And what you have is, if you ever heard somebody with a really raspy voice, it's a condition known as dysphonia, which is loss of voice. And chronic dysphonia occurs over 4 weeks or greater. So lots of people have chronic dysphonia in that category. So what we're hoping to do is go in and be able to treat that scar tissue, because as it thickens, it reduces the mucosal wave, which then diminishes voice. So that's the correlation between scarring and the affliction. So we ran a phase 1 study in this by using these autologous fibroblasts, so from the patient themselves, same process as our current CMC, so no change in our platform for a regulatory. And they're grown up and then re-injected, and then we think there's a remodeling cascade that could be occurring that causes them to, remodel that tissue, and hopefully create some pliability that can create the wave again. And we've seen some of the data from this in phase 1, and we translated that into our phase 2 program.
So maybe touch on that phase 1 data, and if you can, describe the phase 2 that's ongoing.
Yeah, absolutely. So the phase 1 study was a pilot, open label, five patient study, small study, but gave us a lot of information on how to design the further on studies. It was done at at UCLA, and what we designed there was a treatment session where the vocal cords were injected. They're basically injected through the larynx and guided by endoscope. So it's a fairly standard, sounds difficult, but actually for otolaryngologists, a fairly standard procedure for injecting into the vocal folds. They're dosed over 3 treatments per vocal fold. And then we looked at endpoints out to a year with a primary at 4 months in phase 1. And in that, we looked at 2 primary endpoints. One is called the mucosa wave grade, which is essentially a stroboscope that is shined onto the vocal folds. You hold a vowel, and then they can actually judge the grade of wave across the top of the vocal fold. That gives you an idea of how much actual wave you can form. And the second endpoint is called the voice handicap index. It's a published literature subject graded scale that's been around for a long time. And on that, it says you should have about an 18-point reduction in scores and improvement in voice. And we saw that plus. We saw about 28-point reduction. We saw significant reduction in vocal wave grade as well. So that gave us enough confidence to design a phase 2. The phase 2 is a 20-patient, double-blinded, multi-centered study. We're at 3 centers, including UCLA, and then we added NYU and also Stanford University. It's 2-to-1 placebo-controlled, so we are going to get some efficacy look at these data, looking at the same endpoints. And then in our discussions with FDA, they asked us to add another endpoint called GERBOS, which is essentially a expert-rated scale that looks at different attributes of voice. Graveliness, raspiness, they're all different attributes, and is also in the literature as well known. And we're through the injection phase. We're in follow-up now, just completing that, and we're looking to read those data out in the second quarter of this year.
Great, that's really helpful. Just to kind of get a sense of the size of this market, how many Americans or how many people in the developed world where this might make sense are afflicted with vocal cord scarring?
Yeah, it's a good question. This has been one of the more difficult epidemiology exercises we've gone through. Voice tends to be ignored because when you have a raspy voice, you tend to just deal with it. That's actually very common. So we actually had to go to a lot of voice centers. There's about 100 of them in the U.S. So this isn't like your normal ENT that would treat your allergies. Voice centers to really hone in on really what's the epidemiology, and there are millions of people who suffer from dysphonia. However, dysphonia can be caused by paralysis, by other neurological disorders that wouldn't be solved by fibroblast cells. We really wanted to kind of focus that number in on who really suffers from scars. And although there wasn't a specific scar number, we were able to use the available data and MedWatch data to get it down to about 64,000 Americans we think are chronic and really could benefit from being severe or moderate to severe. We think that is probably somewhat ubiquitous around the world. We haven't looked at sort of where the pockets in third world might actually accelerate that. The common theme seems to be teachers. A lot of teacher unions talk about this condition, and accident victims, people who have had different kind of surgeries, in the aged population. That's really sort of where the focus has been.
Many financial analysts from hosting Q&As.
Years of talking.
You mentioned the partnership with Intrexon. Tell us about this alliance and how the two companies are working together.
Yeah, good question. So Intrexon and we sort of met in the middle in an interesting time, and we started our collaboration back in 2012. We had just come off of a BLA, and we're looking at ways to look for more medically relevant. Back then we were looking at more cosmetic indications, but wanted to get more medically relevant side of the business where we had regulatory know-how. We just gone through this process. We knew how to manufacture, and we're planning on ways to scale up an autologous manufacturing platform. And they had this wonderful array of DNA tools that can be used to look at different ways to introduce genes into these cells. So somewhere in the middle, we were trying to figure out, like, where can we really treat diseases? And I'm sure we'll talk a little bit about some of these, but there are some very interesting monogenic disorders in skin that are just very terrible and have pediatric implications. And in the middle of that, they were able to take our fibroblast cells and figure out ways to really efficiently transduce them what I would say are some of the largest genes we'll see transduced compared to some of the other gene therapy companies. And so that's where the collaboration came from and grew.
That's really helpful. And picking up on that, you guys are working on 2 gene-modified products under that alliance. The first is FCX-007 for RDEB. So maybe start off by telling us a little bit about this horrible condition and how 007 works.
Sure. So if you're not familiar, the disease is called recessive dystrophic epidermolysis bullosa. We'll call it RDEB for short. And it's an autosomal recessive monogenic disorder where kids, it's mostly pediatric, are lacking the ability to produce full-length collagen 7 or type 7 collagen. And why that's important is this forms a protein basically called an anchoring fibril. Think of it like in Velcro, the hoops in Velcro. Well, the hoops would be the anchoring fibril, and then they attach to the other side, and then you have to, they make a good bond. Well, in skin, you have something similar. You have an anchoring fibril in the basement membrane zone between layer skins, and it binds to the other side of the epidermis through several protein cascades, and it keeps your layers of skin together. If you have a mutation in this particular gene that forms this protein, your layers of skin end up sort of laying on top of one another, and any lateral friction actually can separate them very easily, and you get blisters. And because the body will sense a void and fill it with fluid, and if you put any pressure on the blister, they spread. So what happens with these kids, they're called butterfly kids because their skin is as gentle, is sort of as fragile as a butterfly's wings. They get widespread blistering, they get a lot of wounding, and where the real issue comes from is these woundings can create contractures and rewounding over the course of their lives and can very easily lead to squamous cell carcinoma, which is fatal to them within 5 years. Also sepsis, as you can imagine, is a very large issue. It also affects the oral mucosa as well, so they go for a lot of surgeries for oral, for dilatations of the esophagus, finger separation surgeries because of all the wounding and rehealing. And the mortality is in the 20s. They rarely make it into their 20s. Right now, care is palliative. It's all wound management. So there's really not a genetic sort of answer for this yet. So how FCX-007 works is we've taken our fibroblast cells, which obviously are in the skin, and they're genetically modified with a gene called Col7A1. It's a 9 kb large construct that encodes collagen 7. The goal being is then we'll take their own cells, we'll introduce this gene, now a working sort of characterized version of this gene, synthetically made, and then reintroduce it back into the skin in hopes to produce this protein and then create these anchoring fibrils again.
Excellent. And we're running out of time. We actually needed more time to talk than just 15 minutes. But the FDA has requested some additional preclinical data for 007 prior to submitting an IND. So what is that preclinical data? What remains to be done? And when can we get the IND?
Yeah, good question. So yeah, we did submit an IND. We got a request to do some additional work. Our previous tox study was a tox pharmacology hybrid that we actually discussed with FDA in pre-IND and executed. We saw no toxicology issues. We reported this separately. Upon review, the reviewers and the FDA felt that they'd like to see that in a separate tox-only study. In that particular one, we had done xenografts into the mouse with RDEB substitutes and then injected those. And this, they took a step back and said, well, let's take one more look at this without any kind of issues looking at the graft and also the product at the same time. Do they compound one another? So we've executed this new mouse study. We're just collecting the data now. We still affirm our end of 1Q filing of the IND. So as you can tell, that's coming up very quickly. And then, obviously we'll resubmit back to the FDA.
Great. So it's going to be a very busy 2Q.
Yes.
So in the 30 seconds we have left, how much cash does Fibrocell have, and how long will that fund the company?
Right. So we announced at the end of 2015 we had $25 million. 29. Sorry, thank you, Keith. Our CFO is here. If any questions financially? $29 million. We opened a new ECC with Intrexon. We're going to be looking at localized approaches for arthritis and joints. So as opposed to some of the widespread systemic biologics, so we're really excited about this new one. That was a $10 million upfront payment that occurred right at the end of the year, and we're projecting that our cash will last into the fourth quarter.
Excellent. Thank you very much, John. Appreciate the time.
Thank you. Thanks, Ted.